Hey everyone, Dr. Jack Order here, and today I'm going to be taking you through inflammatory signaling, right? So the resident immune cells in this situation have already detected the pathogen or damage, so the pathogen-associated molecular pattern, the PAMP, or the damage-associated molecular pattern, the DAMP, and they've detected it on their pattern recognition receptors. So what happens next? Now, I've got this scanning electron microscope image here, and in it we can see holes in the membrane of a macrophage. And I've got that because this is just a really cool image, but those holes were actually intentionally created by the macrophage in order to pump out as much cytokine as possible out of those holes. It couldn't go through conventional signaling pathways, so it was like, crud, I'm infected, I have got to get out as much cytokine as possible. So the macrophage punctures holes in its own membrane. It's really cool. I'm going to cover that later in a much later video, not in this playlist, about what's going on there, but it's such a cool image to see the macrophage commit suicide like that. Anyway, let's jump into it. So yes, we've done the sensing, and now we're going to move on to the signaling. And you can see these two molecules here, prostaglandins and cytokines. Those are the two major ones I'm going to be covering. Note that there are more. There are loads of different families of signaling molecules that induce inflammation, but these two are the famous ones and massively involved in inflammation. Another one is histamine, which I'll touch on. So first, let's look at histamine. So this is you've got to think about this as a mast cell specific thing. Also, basophils do it. Basophils are the circulating version of a mast cell, is a great way to think about it. But the resident immune cell, the mast cell, IgE has bound to a parasite, and that or a molecule secreted by the parasite, a PAMP, and that's bound to the Fc receptor, which causes degranulation of the mast cell. So in those granules is tightly, densely packed histamines, right? Now, a key thing here is the histamine is pre-synthesized. Now, I kind of touched on why that's important. If you want a fast response, you want to pre-synthesize your signaling molecules and then just bang them out when you need to. So if you want a fast response, you need to pre-synthesize your signaling molecules and pack them into vesicles ready for degranulation. Another important point on that is pathogens can attack our immune cells. So to prevent them from doing the signaling, right? The idea is the pathogen wants to shoot the messenger before the message is released. So if we take our time to synthesize all our signaling molecules after detection of a pathogen, there's opportunities for the pathogen to kill the immune cell before it gets the signal out. So by pre-synthesizing our histamine, our inflammatory signaling molecule, and popping it into granules, we are ready to go to release those, that signaling molecule. It's packed into granules, as I mentioned, so it's tightly packed in there, and that distinguishes it as a granular site over a non-granule cell. And that comes more into it with this technique that we use in immunology a lot called flow cytometry that can detect those granules. So we can divide the immune system up into immune cells with granules and immune cells without granules. So histamines in both mast cells and basophils, as I mentioned. Basophils are kind of the circulating version of a mast cell. Now, degranulation is signaled by calcium flux, right? So just quickly, I want you to stop and think, is calcium levels inside the cell normally high or low? Normally. So under a normal situation, where is the calcium? The answer is low. So normally we have high calcium outside the cell, low calcium inside the cell. Some people describe a cell like a banana in a pool of milk which has had table salt added to it. And that's because the extracellular fluid, which in this case is the pool of milk, is high in calcium and sodium, which is in table salt, and chloride, which is in table salt. Inside the cell is high in potassium. So if you imagine all cells are like a banana in a swimming pool of salt, it's a good way to remember where the ions are in the human body. So the banana is low calcium and the milk is high calcium. So the cell is low calcium and the extracellular fluid is high calcium. And one of the important points of this, so Fc receptor activation causes calcium influx, which signals for the granules to fuse with the cell membrane and release its histamine, right? So calcium influx signals that process. One of the great things about that is Fc receptor activation definitely causes that. Boom, fantastic, that's great. So we will release histamine if we get Fc receptor activation on our mast cells. But pathogens produce pore-forming toxins, right? That's because pathogens want to eat us, so they lyse our cells to release the nutrients so they can eat it. So Staphylococcus aureus releases a protein called hemolysin to pop our cells so it can consume the nutrients inside. But also pathogens produce pore-forming toxins, popping toxins, to kill our immune cells, so we can't have an immune response to that. But if you put a pore into a mast cell, if you pop a mast cell, you are going to cause calcium influx and cause degranulation. So it's kind of like a landmine. We've got landmines all throughout our body, and if the pathogen goes too far and pops a mast cell, you're going to get automatic degranulation and histamine release. So it's a fail-safe. So you don't necessarily need FC receptor activation. It can actually just happen through a pore-forming toxin released from a bacteria. Very clever. It's just a landmine. If you attack me, I'm ready to go. If you try and pop me, I will release my histamines automatically because they're pre-synthesized and all I need is calcium influx, and that will happen naturally if you pop a hole in my membrane. Super clever. Super clever. So now let's compare that to say a macrophage that's had a PAMP or a DAMP activate a cell surface receptor here. We're going to get the release of prostaglandins and cytokines. So we're not going to get that histamine release. We're going to get prostaglandins and cytokines. And a subset of cytokines is called chemokines. So all chemokines are cytokines, but not all cytokines are chemokines, and I'll discuss the difference there just briefly. So prostaglandins, first thing is they're a small molecule. In biology, we often break things up into small molecules and large molecules, and largely that means large molecules are peptides or proteins, and so that is chains of amino acids. They're very large. We measure those in kilodaltons, so thousands of daltons, so that very large molecular weight. Small molecules are often just a few cyclic rings of carbon or a little chain in this case, because these are lipid-like, and they're normally, in the hundreds of daltons. So they're way smaller, and they're not sort of made up of a chain. They're just a single molecule. They're not a polymer. They're not a chain of repeating units like amino acids or complex carbohydrates. They're just a single small molecule. And there's certain advantages and disadvantages to being a small molecule. It's important. So prostaglandins are small molecules, and they're produced by an enzyme called a COX-2 enzyme, which results in the enzymatic digestion of the phospholipid membrane. So there are lipids in our membrane, and our cell just uses that as a source of lipid molecules. So if you need sort of a hydrophobic lipid molecule, you borrow some of the lipid from your membrane, digest it down into a signaling molecule, and send it out as a signaling molecule. So it's turned on, and COX-2 is an enzyme that's not normally expressed in the cell, and it's turned on in response to DAMP or PAMP detection. So we get activation of our pattern recognition receptors by a DAMP or a PAMP. The COX-2 enzyme is turned on, and it is slow and capable of prolonged signaling. So there's another little point I want to cover here is the barriers of the human body are made out of lipids, membranes, right? So the barriers of the human body are made out of membranes. So there's a barrier between our blood and our brain, for example, that's made out of membranes. There's a barrier between inside our blood and outside our blood that's made out of cells, and cells are surrounded in a phospholipid membrane. So membranes are the barriers of the human body. But if you make a signaling molecule out of a lipid, lipids are soluble in lipids, and a membrane is a lipid, a phospholipid membrane, right? So membranes are soluble in membranes. What that means is the barriers don't work, right? So the lipid is able to flow in every direction nice and evenly. And we know this when it comes to drugs. So a lot of drugs that target the brain are lipophilic because they need to get through that barrier. So if you think about cannabis oil, it's an oil, it's a lipid, it can easily get through the barriers of our body into our brain. So the body knows this too, and so when it produces prostaglandins as a lipid, it can diffuse in every direction, act as a local signal of inflammation. There are no barriers to it, right? So it's capable of slow and prolonged signaling in a very local area with barriers are no issue. It can spread out evenly through it because it's soluble in those lipid membranes of the cell, which is very cool. So that's why it's created from our phospholipid membrane. Think about it. It took a piece of our phospholipid membrane, we changed it to a prostaglandin. Now it's soluble in the membrane. That's where we got it from. So now it can flow through all the barriers of the human body. Now, non-steroidal anti-inflammatory drugs like aspirin, ibuprofen, Voltaren all inhibit the COX-2 enzyme. So they inhibit the production of prostaglandins. And that's how they work. And that actually tells you a little bit about another function of prostaglandins, is they help the signaling process of pain. So it's a local pain response. You can imagine it kind of swells out in every direction around that location because it's lipophilic and it acts locally around it, spreading out, creating a zone that's high in concentration of prostaglandins, causing pain and inflammation. And so if we inhibit the COX-2 enzyme, we can block pain and we can block inflammation by inhibiting the production of prostaglandins. So we need this gene expression to occur after response to the pattern recognition receptor to turn on the COX-2 enzyme, which then creates prostaglandin molecules. And I've just got one of the many examples here. So prostaglandins are a family, so don't talk about them as a single molecule. They're a family of molecules that cause these responses. Now let's compare that to cytokines. Cytokines are proteins. So they're a polymer made up of chains of amino acids, right? So they're not a small molecule, and we measure them in the thousands of Daltons, right? So they're very heavy, large molecules. They too are slow and capable of prolonged signaling, and they're massively varied. So this is one of the great things about proteins, is you can tweak the sequence to dramatically change their structure. And so there is a huge diversity of structures, which results in a huge diversity of function. So they're both capable of inflammatory and anti-inflammatory signaling. It depends on the cytokine. There are loads of cytokines to choose from. And I'm not going to cover them all. I mean, I don't think there's a biologist on the earth that understands what all the cytokines do, but there are a few famous ones that I will cover. Now, an important point here is proteins are typically water soluble, right? So are often water soluble. So this isn't going to flow through the barriers like the prostaglandins, but it is going to stay in the blood. Once the cytokine gets into the bloodstream, it's going to stay in that water soluble environment of the blood, so it can do much longer distance signaling, which is fantastic. So it will be cytokines that tell the immune cells in your bone marrow to come out and go into the bloodstream, because we need to up our immune response, our innate immune response. So cytokines are capable of slow and prolonged signaling as long as we keep producing that cytokine, and long distance signaling as well. And chemokines are a subset of cytokines, and they're involved in chemotaxis. So that's a great way to remember it. So cytokines that have a chemotaxis component are called chemokines. So they signal to the immune cells to go up the concentration gradient to find the location of tissue damage. And a good one is MCP1, which is monocyte chemoattractant protein 1. So MCP1, monocytes will climb up the concentration gradient towards that. So how are they produced? So we need a pattern recognition receptor activation, which causes a change in gene expression, often NF-kappa B is that transcription factor, and that causes the release of the cytokine. Sometimes that cytokine requires post-translational modification like IL-1 and TNF. Now that's because these are such potent inflammatory cytokines, we need two checks. We can't just go willy-nilly releasing these cytokines. So we need this pattern recognition receptor activation, and we need a second signal that will lead to post-translational modification. So we need two signals. It's like if you've ever seen on the movies, they have two keys that you have to turn before you release a nuclear missile, and you have to turn them simultaneously. So it's kind of like that. The cell has 2 points in which these cytokines could not be released. So they could not be turned on in the first place, and they could not be post-translationally modified. That's because they're so inflammatory, you better be sure that you know what you're doing before you release them. But there are other ones that are just released straight away, like IL-6, which is a much more mild cytokine. So here are some cytokines here: IL-6, IL-1, and TNF. Again, the acronym isn't hugely important, so everyone just calls them IL-6, IL-1, and TNF. And just so you know that there are drugs, just like the COX inhibitors help block inflammation, there are drugs that block cytokines and are really, really beneficial for some patients with chronic inflammatory diseases. So there's an anti-TNF drug that's effective for rheumatoid arthritis, for example. So there are many drugs that block many of these signaling pathways and are very effective at treating diseases caused by inflammation or pain. So let's have a look at those chemokines. So here we have a resident immune cell, and it is releasing a chemokine. So the highest concentration of that chemokine is going to be directly around that cell, and then it's going to dissipate, and we end up with lower concentration. And here we have, say, a monocyte. It's going to climb up that concentration gradient as a positive chemotaxis. So that's why they're called chemokines. And this is essentially how immune cells get to where they need to be. First, there's cytokines that tell them get out of the bone marrow, get activated, get ready. And then there are the chemokines that say, okay, come here, come to this location specifically, because this is where the pathogens or damage is happening. So next we're going to have a look at that inflammatory response. What happens once those immune cells get there? So we've looked at the sensing, we've looked at the signaling. Next we're going to look at what happens to the immune cells once they get there.